Actin slides over myosin, and this is how muscle contracts. And this is a muscle that is made up of multiple muscle bundles or muscle fascicles. Here I am taking out one muscle bundle or muscle fascicle, and that is again made up of multiple muscle fibers or muscle cells. How is the Joe, students? If you want to move like this or want to dance like Michael Jackson, you have to use your skeletal system as well as muscular system. So if this is the hand of your body, if you remove the epithelial tissue, you're going to get the muscles, part of muscular system, and these are associated or present on our bones. Now, if you take one single muscle from your body that is made up of multiple muscle bundles or fascicles connected by connective tissue called fascia. Now if you take out one single muscle bundle from it, there is one fascicle that is made up of numerous muscle fiber or muscle cells. Now if you take out one muscle cell from it, that looks striated or striped because these are voluntary striated skeletal muscle. Now you want to find the reason behind this striation. Now you have to check inside this muscle fiber, and you will be getting this kind of parallelly arranged fibrillar structure inside every muscle cell called as myofibrils. Now here you can see inside myofibrils, myofilaments, special proteins and fibers are present in a proper manner that is giving the dark light or the striated pattern. Now if you get inside those myofibrils, you are going to get this banding or striation pattern due to the special arrangement of thick filament and thin filament. Now these blue filaments, blue lines are thin filaments or actin, and the red line is thick filament or myosin. When inside your body muscle is contracting, and if you follow the sliding filament theory, they are moving like this, sliding upon each other. Yes, these actins are sliding upon myosin. Now here you can see one sarcomere is given here, that is the length in between two zigzag line or Z disc. And these blue lines are actin filaments, and the red line, that is myosin filament or thick filament. When these actin filaments are moving towards the central M line or sliding over myosin, gradually the length of sarcomere is decreasing because the Z lines are coming closer, and that is called muscle contraction. When you're talking about muscle contraction, we can start like this: start the motor, bring the action, let's become Michael Jackson. Yes, our muscles get nerve impulse or motor signals from our central nervous system. Upon getting that, action potentials are generated inside those muscle cells, and upon generation of action potential, they start contraction. They start contracting and relaxing. That is called contractility of muscle fibers. And yes, it's due to your contraction and relaxation of your muscles, you can dance like Michael Jackson, you can walk, you can talk, or you can run. We are at PW NEET English. Welcome back again to another session of one shot on locomotion and movement, and the topic is muscular system. Here we learn with fun. Myself Biswajit, your zoology master. And after completion of this chapter, do not forget to check our PW app. The link is just given here in the description box. Upon clicking the link, you'll be heading towards a DPP present inside that PW app, and you have to solve it to boost your knowledge, to check your learning outcomes. And we are also with a PW NEET English WhatsApp channel. You'll be getting the QR codes here. You just have to scan it, and that will lead you towards that WhatsApp channel, and there you can get everyday notifications, update, new videos, prompts, polls, questions, and many more. So what you are waiting for? Let's jump in. Yes, in this chapter, these are the topics we are gonna cover today, and the topics are obviously muscular system. We are talking about muscles, locomotory structures into different animals. We're going to talk about the classification, different types of muscle. There are skeletal muscle, cardiac muscle, smooth muscles, and obviously you will be detailing on skeletal muscle. We'll be talking about how a single muscle fiber or a muscle cell looks like, types of proteins involved in the formation of muscle fiber and responsible for the contractility and elasticity of a muscle fiber, and inside sliding filament theory given by Huxley, two German scientists. We will be studying the whole mechanism of muscle contraction, and we will be concluding this particular one shot with few muscular disorder. So let's start. Okay, so we can say, or we can introduce the chapter like this: all locomotions are movements, but all movements are not locomotions. Yes, that means locomotion, it's a broader aspect, and movements are part of the locomotions. Now what is difference? Now movement means there will be no change in position, or there will be no displacement of a living body. So here I'm just standing, I'm standing in a static position, but I am moving my hands, I'm talking with you. So that's a kind of movement performed by the muscles. Yes, but here I am changing my place, displacing my body. So whenever I am displacing my body or changing my place from one position to another position, that is called locomotion. While I am static, here also I am showing movement. While I am locomoting, here also I am showing movement. So that's why in static position, we will be calling this kind of change of posture of my body as a movement, and in kinetic position or in locomotion, when I am locomoting, we will be calling that I am changing the place, but that phenomena is also involved with multiple types of movements, and that's what it's trying to explaining. Okay, so it's simply like this. Suppose this is our friend Chintu is standing in a place and is doing gym with a dumbbell. So let's put a dumbbell in the hand of Chintu. We'll be moving the hand to and fro with the dumbbell. So that's simply a kind of movement. Now let's convert these movements into locomotions. Now suppose Chintu was calmly in a peaceful environment. He was doing gym without changing his position, without displacing his body. But suddenly he saw an ox or a bull is coming, a red bull is coming, and whenever he will see that a red bull is coming towards him, now that will give him a wing and he will run away. So he is running like this. He is running like anything. So the phenomena of changing the position from point A to point B, that is locomotion. And obviously you can see he's moving his hands and legs, but here while he's performing just movement, he's only moving his hand to lift off the dumbbells towards up and down. So here we can differentiate in a detailed manner the differences of movements and locomotion. So it's written here, movements results in change of place is locomotion, and most of the locomotions, they are voluntary activity. Yes, by our will and wish, if we are performing a particular work, so that will be called as voluntary activity. Now let's differentiate between movement and locomotion. So first with few examples. So it said that streaming of protoplasm. Yes. Now we can observe this kind of streaming of protoplasm during the prior to perform phagocytosis by phagocytic cells or amoeboid cells. Movement of cilia, flagella or tentacles, like movement of cilia, ciliated cells present into our ciliated epithelium, present into our tracheal wall, present into the inner wall of fallopian tube. Tentacles, hydra will catch the prey with the help of their tentacles. Jellyfish uses their tentacles to catch their prey. Human, we use our limbs, means hands and legs, jaw, eyelids and tongue. Like while I'm talking, I'm utilizing the muscles of my tongue and jaw for movement. When I'm blinking my eyes, I'm using the muscles associated with eyelids for the movement. Limbs, while I'm eating, while I'm writing, I'm using muscles associated with my forearm to write something, to move my hand. Okay. And locomotion means obviously change of place, and most of the times organisms perform locomotion in search of food or shelter or mate or to find breeding ground. You may have heard about the term migration. Few mammals or birds are migrating thousands of miles in search of food and breeding ground. Few birds are coming to India from Arctic region or Antarctic region to get better climatic condition or to escape from their predator. So these are the reasons mentioned in the NCERT that why organisms will be performing locomotion. Okay. Now while you are performing locomotion or I am performing locomotion, it's a coordinated activity. Locomotion is a coordinated activity because the signal will be generated from our neural system. Neural system is responsible for control and coordination and to sense the environment present inside our body as well as present outside our body. Then neural system will be giving signal to muscular system. Muscles will show contraction and relaxation or extension. Muscles have contractility, extensibility, elasticity. So from neural system, the message will reach towards the muscles, and when muscles are exhibiting contraction and relaxation, they will transfer the force within the joints of our skeletal system. So ultimately skeletal system, the bones or the joints present in skeletal system are also involved in the process of locomotion. So locomotion, that's why it said that it's a coordinated activity of our neural system, of our muscular system and our skeletal system. Okay. So when you study the diversified animal kingdom, you may find different types of locomotory structure present into different organisms. First, we will be talking about paramecium. So here in paramecium, you can observe cilia. Now, what are cilia? They are nothing but extension of the cell, or better to be said that this cilia are their extension of the cell membrane. Okay. Now, movement performed by cilia is called ciliary movement, and particularly the cilia are called as kinocilia. They are responsible for kinetic activities or movement. Kinocilia. Another type of cilia you may find into animals, they are called stereocilia. They are static in nature. They are not involved in the process of movement. And this kinocilia, they're originated, or they are just extension of their cell membrane. Yes. Now here in this particular paramecium, you can find here this mouth-like structure present into cell. The mouth of cell, you may call it cytopharynx. Cytopharynx. And this cytopharynx is surrounded by kinocilia. By the movement of this cilia, the ciliary movement, it is creating a vortex inside water and intaking the food particles present into the water inside the cell. Right. Now let's talk about the movement of amoeba. The movement exhibited by amoeba or other phagocytic cells with the help of pseudopodia, false feet. Pseudopodia. Pseudo means false, or pod, that means feet. So while they are performing phagocytosis, the cytoplasmic extension will form these false feet to grab their prey. So while they are performing phagocytosis, basically with this pseudopodia, with the help of their false feet, they are performing phagocytosis. What do you mean by phagocytosis? When a cell is engulfing something with the help of cytoplasmic extension or pseudopodia. That process is called as phagocytosis. Yes. Now let's come to another organism, Euglena. For their locomotion, they have flagella. And the movement will be called flagellar movement. Flagellar movement is also exhibited by spermatozoa or sperm. Hydra. Let's talk about the movement of Hydra. They'll be performing movement with the help of these tentacles. These tentacles are equipped with neurotoxic stinging structure filled with neurotoxin. These tentacles have these stings called nidoblast. Right. Okay. But here we are talking about the locomotion. And with the help of these tentacles, they can catch their prey like small organisms hovering nearby them. And also Hydra can perform locomotion. And how they will be performing locomotion? Though they are kind of sessile animals, sessile cnidaria, but sometimes with the help of tentacles, they try to perform back volt or somersaults. The movement, the locomotory movement of Hydra can be called as somersault or backward movement. Okay. Now we have classified different types of movement into animals or into our body. First, we will talk about the amoeboid movement and how amoeboid movement, and what do you mean by amoeboid movement? The phagocytic movement performed by amoeba or amoeba kind of cells like phagocytes present into our bloodstream, who are phagocytic cells. They are neutrophils, the primary phagocytes of our body, and macrophages or monocytes. They are the phagocytic or amoeboid cells. They perform amoeba kind of movement with the help of cytoplasmic streaming and by forming pseudopodia. So, they will perform the movement by streaming of their protoplasm, right? And also microfilaments present inside their cell will help in the process of this protoplasmic streaming. And few of the leukocytes, not all leukocytes, leukocytes means WBC, few of the leukocytes will perform such kind of amoeboid movement. Now, who are they? We call them phagocytic cells. Phagocytic cells or amoeboid cells. They are macrophages and neutrophil. Now, question can be asked that who are primary phagocytes? Neutrophils are primary phagocytes. What do you mean by that? After an infection in our body, the first cells will appear or arrive to see to tackle the situation that infected place. They will be neutrophils, and obviously they are the most abundant WBCs in our body. Upon the arrival of neutrophils, macrophages will come from the bloodstream, and macrophages can be also called as monocytes while they are residing in bloodstream. So macrophage and monocytes, they are same cell. We'll be calling them monocyte inside the bloodstream. And to fight, to check an infectious agent, when those monocytes are leaving blood vessels and entering into the tissue space, we call them macrophage, right? Okay. So this was all about amoeboid movement. Now you need to remember amoeboid movement, how amoeboid movements are performed by cytoplasmic or protoplasmic streaming, and secondly with the help of microfilament. Okay. Next come to ciliary movement. Specifically we are talking about kinocilia. We are not talking about stereocilia. Now, what is the difference between kinocilia and stereocilia? Already I said that kinocilia are responsible for the ciliary movement. Stereocilia, they are kind of a static structure. Cilia or kinocilia, they are extension of cell membrane mainly, and stereocilia, they are extension of protoplasm or extension of cytoplasm mainly, like microvilli. Okay. Now, where we can get cilia or this kinocilia inside our body? They are present into the internal tubular organ, and the lining is made by the ciliated cells called ciliated epithelium. And in structural organization chapter already you have studied that ciliated epithelium can be found in trachea, in fallopian tube, and also inside the ventricles of our central nervous system, the ependymal epithelium. Okay. Now few say that ependymal cells do not have kinocilia. Ependymal cells are equipped with stereocilia. Okay. But for sure we can say the inner wall of trachea and inner wall of fallopian tube are equipped with kinociliated epithelium. What is the function of ciliary structure, ciliary epithelium inside trachea? The ciliary movement helping the inner wall of trachea to channelize the air and dust particles from one direction to another direction. They are filtering the dust particles and channelizing the air during the time of inhalation from outwards to inwards, during the time of exhalation from inwards to outwards. What is the function of ciliary epithelium inside fallopian tube? Inside the fallopian tube, there will be movement of gametes. Now, upon the process of ovulation, the secondary oocyte enters into the fallopian tube or oviduct. Now, we know that secondary oocyte or the immature ovum is not motile in nature. Spermatozoa, they are motile in structure. So, by the swimming process helped by the flagellar movement, sperms will be reaching to the ampulla or ampulla isthmus junction and waiting for the ovum to arrive here. Now, how ovum will be arriving? By the process of ciliary movement present into the ciliated epithelium of fallopian tube. Now, let's talk about muscular movement of our body, movement of our jaw, eyelid, esophagus, the peristalsis movement occurring inside our alimentary canal with the help of special smooth muscles, movement of our limbs even while you are chewing or while you are talking, the movement of your tongue with the help of muscles. And few examples are running, walking, talking, blinking, all are result of muscular movement. Now let's talk about flagellar movement. Yes, spermatozoa or sperms, they have motile flagella, right? Now with the help of this motile flagella, the sperm will be swimming. The sperms will be swimming towards the ampulla region of fallopian tube just after the process of insemination. So when you talk about flagellar movement, we need to take the names of spermatozoa produced by the testes inside the seminiferous tubules of testicles. Now let's talk about muscles. So most of the muscles of our body are mesodermal in origin. I am saying mostly. Not all the muscles are mesodermally originated. There are few muscles who are originated from ectoderm. So here I'll be talking about ectodermal muscles. Yes, they are present into our eyes and the name is iridial muscle or muscle of iris and muscle of ciliary body. These two muscles are ectodermal in origin. So that's why be careful when you are reading a statement. If it said that all the muscles are mesodermal in origin, so that's going to be a false statement. Okay. Muscles. If we consider the body weight and compare with muscular weight, they contains 40 to 50% of our body weight, though muscles are highly rich in water particles. Now let's talk about few important properties of muscle. Okay, and the properties are excitability, contractility, extensibility, elasticity and conductivity. These are the properties of muscles. Now you will be finding similarities with nerve fibers or neurons with few of the properties of muscles. What are they? Excitability. You will be finding in both the neurons and muscle cells, they have excitability. Now, what do you mean by excitability? Upon giving a stimuli or stimulus, when it's beyond the threshold level, the muscle will start responding against it. That means if you are giving motor signal to a muscle cell, it will show contraction or relaxation or extension. So that is called excitability. Now, what is conductivity? That property is also you can find into muscle fibers and into neural fibers or into nerves or neurons. Conductivity. This is called action potential. Inside the neurons, we'll find nerve impulse is getting propagated, and inside the muscle cells also you'll be finding similar kind of propagation of action potential. So that's why we are saying that excitability and conductivity, these two are common criteria present into neurons as well as muscle fibers. But muscles have something extra. And what are they? Contractility, extensibility and elasticity. Now let's elaborate what is contractility, extensibility and elasticity. Now, suppose this is the normal condition of your hand. You are standing in such a posture. So this length of your muscle will be called as the relaxed state. Now, if you are doing such, if you are moving your hand towards your body, so gradually what will happen? The width of your muscle will increase, but the length will decrease. So this sort of shortening of length of muscle is called contraction. Now once you are contracted, you can go back to the original position. You can your muscles can gain back the original position, and this condition is said as you are relaxed or your muscles are relaxed. So what is relaxation? Gaining back the original length of muscle. That's called relaxation. So you can perform such contraction and relaxation. Contraction and relaxation. Now let's go toward extension. Now you are stretching beyond the normal limit. You are lengthening it. You are stretching it. Now this kind of activity can be called as extensibility or extension, that is stretching or lengthening your muscles beyond its normal limit. When your muscle is stretched, that capability is called extensibility. And after performing extension, a tendency will be there to gain back, to regain its original length, that is called elasticity. Okay, now we'll talk about types of muscles. We'll classify muscles into three groups. What are they? First, skeletal muscles or striated muscle. Second group, visceral muscle or smooth muscle. And third group, cardiac muscle. Now, what is the basis of the classification of muscle? Okay, these points are also written here. Basis of classifications of muscle. First, location. Like if you talk about skeletal muscle, they are present upon a skeleton associated with a skeletal system. That's why they are called skeletal muscle. Visceral muscle present into the viscera, that means internal organs. Cardiac muscle present into your heart. That's why you are calling them cardiac muscles. Next criteria to classify muscles: appearance, how they are looking. If you are getting a muscle with lots of dark and light bands or stripes, you may call it striated muscle. If you are getting muscle, this cytoplasm is very much clean, there is no banding or stripes are there, you can call it smooth muscle or unstriated muscle, right? So the classification is also based upon their appearance. This cardiac muscle, they will also show striation, but they are branched in nature. We'll be detailing about them. So cardiac muscles and skeletal muscles, they are striated in nature. And obviously the third criteria is regulation. Either they are regulated by your autonomic nervous system, or they are regulated by your somatic nervous system. If your muscles are regulated by somatic nervous system, we will be calling them as voluntary muscles. They are performing contraction, relaxation based upon your will and wish. And if your muscle cells are not listening to you, your will and wish cannot control them. That means they are under control of autonomic nervous system. We will be calling them involuntary muscles. So, classification of muscles are based upon three criteria: their location of presence, secondly, their appearance, either they are striated or not, and third, their nature of regulation, what kind of nervous system is regulating them, either they are under voluntary control or involuntary control. So we'll start with skeletal muscle, as because they are present upon skeleton. So here you can find bone. So they're associated with bones or skeleton. That's why we are naming them as skeletal muscle. Whenever you are studying them, if you enlarge these muscles and take out one single muscle fiber, they look like a cylinder with dark light banding. Due to this arrangement of a specific pattern of myofilaments. So because of the presence of this sort of bandings, we are naming them as striated muscles because of presence of stripe. These are stripe. Light, dark part, light band. Dark band, light band, dark band, light band, dark band, light band. They're under control of somatic nervous system. That's why they are also called as voluntary nerve, voluntary muscles. So you can call them, the skeletal muscle, as voluntary striated muscles or striated voluntary muscles. Appearance of each muscle fiber is like a cylinder. So the shape of the cell is cylindrical, and obviously you'll be finding multiple nuclei inside the cylinder. So that's why they are multinucleated cells, or they are kind of syncyte or syncytium condition is observed into them. Now the question arises here, why they are multinucleated? Okay, for that you need to learn their origin, the birth of skeletal muscle cell. The precursor muscle cells are called as myoblasts. What do you call them? Myoblast cells. Now, if you observe a single myoblast cell, like other cell, it has only one nucleus inside it. Now, if there are multiple myoblasts associated with each other, while maturation or the formation of skeletal muscles were going on inside our body, with time gradually they will exhibit fusion of their cell membrane. So they will exhibit fusion, they will get merged with each other. Now we'll ask, sir, why these precursor myoblast cells are getting fused with each other to share their resources in order to survive, and the resultant cells, they produced this elongated cylinder with multiple nuclei nucleus inside them. That's why skeletal muscle fibers or skeletal muscle cells are multinucleated in nature. Now here you need to remember this: muscle fiber equals to muscle cell. One muscle cell can be called as one muscle fiber. Okay. They have multiple peripheral nucleus. So surface nucleus, or they can be said as peripheral nucleus, and they help in locomotory action and change in posture of our body, like muscles of your limb, muscles of your hind limb or legs. They help in the process of running or movement. So that's why we are saying locomotory action when you are performing running, movement, or swimming, cycling. This kind of skeletal muscles voluntarily helping you to perform. Now let's come to visceral muscle. Now the location, inner walls of hollow visceral organs, like for example inner wall of stomach, inner wall of esophagus, inner wall of intestine. You know about peristalsis movement or churning movement of our stomach and alimentary canal, that is due to the presence of smooth muscle or visceral muscles. And viscera means internal organs. Viscera means internal organs. Okay. You can see the cells. There is no striation. That's why we can call them smooth muscles. They are regulated by autonomic nerves. Regulation is by ANS. That's why they are involuntary in nature. Each cell has a single central nucleus, so that's why they are uninucleated. Shape of the cells are like spindle or fusiform. You can say this shape as fusiform or tapered shape. They help in the process of transportation of foods and gametes by performing peristalsis or peristaltic movement. And obviously the movement or the contraction of these muscles are comparatively slower than the skeletal muscle, so you won't be finding that they are getting fatigue. What do you mean by fatigue? Upon recurring contraction and relaxation, muscle fibers will deposit or produce lactic acid inside them upon performing anaerobic respiration. If a muscle cell is performing anaerobic respiration, it will accumulate lactic acid, and that can cause muscle cramp as well as fatigue of muscle. So if a muscle fiber is contracting and relaxing very fastly, rapid contraction, the muscles with rapid contraction and relaxation will tend to get more fatigue. The muscles with slow contraction will get fatigue rarely, right? So that's why rare fatigue is seen into these visceral muscles, and the reason is their slow contraction. But if we are talking about skeletal muscle, fatigue is seen because they contract very fast. They show fast contraction. Now let's talk about cardiac muscle, muscle present into our heart. In the muscular layer of our heart, that is called as myocardium. Structural muscles and also nodal tissue of our heart like SA node, AV node, bundle of His, Purkinje fiber, they're also kind of modified cardiac muscles. Okay, so cardiac muscles are of two types, structural and nodal or conducting. Structural cardiac muscles, they're present into the wall of heart, call them myocardium. The nodal or conducting cardiac muscles, we call them SAN, SA node, AV node, AVN, AV bundle, Purkinje fibers. These are examples of nodal tissue or modified cardiac muscles. Okay. So, if you observe a single muscle cell, you will find branching pattern, branched muscle cells. This is a single cardiac muscle cell. And you can find striation into that cell. So, branching pattern is seen. They are kind of striated muscles, but involuntarily striated. So, voluntary striated, skeletal muscle. Involuntary striated, cardiac muscle. Though they are branched, but only single nucleus can be observed. So, they are uninucleated cells, and obviously they are rich in mitochondria. They're rich in mitochondria, so they will never become or they will never get fatigue from the 21st week of your embryonic development till death. Sometime after the brain death, your heart is beating inside your body. Your heart keeps beating inside our body. So there is no question, there is no chance it can get fatigue. If cardiac muscles is getting fatigue, that means you tends to get a cardiac arrest or minimally myocardial infarction or myocardial ischemia. Okay. Now, one more important thing, at the junction of this branched cell, you can observe gap junctions to transfer signal from one cardiac muscle cell to another cardiac muscle cells, and these gap junctions are called as intercalated disc. The special or exclusive feature of cardiac muscle cells, and they are nothing but gap junctions. What do you mean by gap junctions? They'll be forming cytoplasmic bridge in between cardiac muscle cells with the help of special proteins called connexin. So you need to remember this exclusive feature of cardiac muscle, intercalated disc. Okay. Now let's move forward. Now we have to study the structure of muscle in detail. So let's understand the organization of the structure present inside each skeletal muscle. Yes. Now we know that the skeletal muscles are attached or associated with bones. So one of the bone is here with us, and this is the muscle. Okay. Now we need to attach, we need to form an attachment of this muscle with the particular bone, and for that we need a dense irregular kind of connective tissue, dense regular kind of connective tissue, that's a fibrous tissue, also can be called as fibrous tissue, particularly white fiber, we call them tendons. They're responsible for joining muscles to bone. What kind of tissue are they? They are dense, regular, fibrous connective tissue. DRFCT means dense, regular, fibrous connective tissue. Okay. Now, inside a muscle, you will find a single muscle is made up of multiple muscle bundles or fascicles. So many fascicles are present inside the muscles. Now, what do you mean by fascicles? Fascicles means muscle bundle. Now, what do you mean by muscle bundle? Muscle bundle means bunch of many muscle cells or muscle fibers. A bunch of many muscle cells. So, muscle has fascicles. A fascicle or muscle bundle has many muscle cells. If you get inside every muscle cell or muscle fiber, muscle cells can be called as muscle fiber, you will get parallelly arranged myofibrils. Inside a muscle cell, you will be getting multiple myofibrils parallelly arranged. That's particular arrangement giving them the striated or banding pattern. Now, inside, if you get inside the muscle myofibril, then you will find myofilaments. Yes, the surface, if you observe the visibility of the vision of the myofibril, then you can find they're finely striated with dark and light bands. We call them I band and A band, isotropic bands and anisotropic bands. Now, this banding pattern or the formation of these bands or stripes due to the presence of myofilaments. Now, who are myofilaments actually? They are actin proteins, will be forming actin filaments and myosin filaments. These are called as myofilaments. This is the organization of a single muscle made up of multiple muscle fascicles. Inside a fascicle, you will be getting muscle cells. Inside muscle cell, you will be getting myofibrils. Inside myofibrils, you will be getting myofilaments. And these myofilaments are basically particular arrangement of actin and myosin. Now, how they are connected to each other, like when multiple muscle cells are forming muscle bundles and these muscle bundles are forming muscles, we need dense irregular connective tissue or tough fibrous irregular connective tissue, and we call them fascia. Fascia is a special kind of connective tissue, dense irregular tough connective tissue, which will be holding these units together to form a compact muscle. Now, let's observe. Suppose this is the muscle and you will be finding a covering of this muscle, and this immediate covering is called as epimysium. Inside that muscle, you will be getting multiple muscle bundles or fascicles connected by fascia. You need to take out one single muscle bundle or single fascicle. So you have taken out one single fascicle, and this fascicle is also surrounded by a special kind of tough covering of connective tissue, and that is called as perimysium. Now get inside one single muscle bundle, what you will be getting? Multiple numbers of muscle cells or muscle fibers connected through fascia, binded together by fascia. Now here you have taken out one single muscle fiber to check what are the components present inside one single muscle fiber. So this is a single muscle fiber or muscle cell, and that also has a covering, and we call these as endomysium. Right? Now peep, inside that you'll be finding multiple myofibrils. You have taken out one single myofibril out of the muscle cell. These are parallelly arranged. So this is the myofibril, and dark and light bending pattern is visible very clearly on the surface of this myofibril, and who are responsible for this dark and light bending pattern for these stripes? Myofilaments. And who are these myofilaments? They are actin and myosins. Now here you can ask me a question, sir, this kind of bending pattern or striation is seen only in striated muscles and cardiac muscles. Because of this, the special arrangement of these myofibrils and the myofilaments or actin or myosin inside that. And for them, there will be contraction and relaxation, means contractility of the muscle is boon of these myofibrils or myofilaments. But we have studied about smooth muscles or visceral muscles. They also have this similar kind of contractility and extensibility. But what is present inside them? Because they are quite smooth, they are quite non-striated or unstriated. So uh, they don't have any kind of myofibrils or myofilaments inside them. The answer is the smooth muscle or visceral muscle cells or non-striated muscle cells, they also have these myofibrils and myofilaments, but they are not parallelly arranged. So this striation is basically due to the special arrangement of this actin and myosin and the parallel arrangement of myofibrils. But in case of smooth muscle cells or non-striated muscle cells or visceral muscle cells, they look smooth because myofibrils are not arranged in a parallel manner. So that's the difference. Rest, their mechanism of action is quite similar. They also do have the myosin and actin filaments. Okay. So let's talk about the coverings of muscles, covering of a single muscle, covering of a muscle bundle or fascicle, covering of a muscle cell or muscle fiber. Covering of muscle is called epimysium. Covering of muscle bundle is called perimysium. Covering of muscle cell is called endomysium. Okay. Now we'll talk something in detail about the muscle fiber or skeletal muscle fiber or skeletal muscle cells. So, can you tell me what you call the study of muscles? It's called as sarcology or myology. Now, what is the meaning of sarcology or myology? The study of fibers, and sarco or myo, the word came from Latin word or Greek word, and the meaning is flesh. Yes, so the flesh of our body is made up of mainly muscles. The plasma membrane of muscle cells are called sarcolemma. The cytoplasm inside muscle cells are called sarcoplasm. Endoplasmic reticulum of muscle cells are called as sarcoplasmic reticulum, and you need to remember this, that this sarcoplasmic reticulum, they are rich in calcium, storehouse of calcium ions. And this calcium will be helping in the process of muscle contraction. The question can be asked that which ions are responsible for muscle contraction? So, firstly it will be calcium ion, and secondly it will be magnesium ion. For muscle contraction, you need two ions, and they are calcium ion and magnesium ion. Remember, these questions are asked on this topic. Okay. Now here you can see a term given as T tubules. So T tubules, they are nothing but they are part of the cell membrane or the sarcolemma of muscle cell, and these particular parts are rich in ion channels. Now if you get inside the sarcoplasm or the cytoplasm of a muscle cell, you will be getting some parallelly arranged filaments in the sarcoplasm, and simply we call them myofibrils. And inside the myofibrils we have those actin and myosin, and they will form particular units in particular interval. We call them sarcomeres, and they are responsible for muscle contraction. So it can be said like this: this myofibrils are actual functional units of muscle fiber. It's due to the presence of myofibrils and the sarcomeres inside them, there will be contractility or the extensibility of muscle cell or muscle fiber. And inside the myofibrils, you will get alternate dark and light banding pattern. Okay. Now these dark bands are known as A bands, and these light bands are known as I band. A band means an isotropic band. Multiple number of proteins are present into this A band. That's why it's called as anisotropic band. And only one kind of proteins you will be finding inside the I band. That's why it's called as isotropic band. The lighter zone are called I band. The darker zone are called A band. Remember this. Now here a diagram of muscle cell or muscle fiber is given. Muscle fiber or muscle cell is given here. So let's check this. So obviously the duty of muscle cell is to perform contraction and relaxation and help in the process of movement and locomotion. Now it needs a lot of energy, and energy currency ATP is produced inside the powerhouse of cell, that is mitochondria. Yes. So muscle cells or myocytes are rich in mitochondria. Kuli powerhouse, right? Okay. Next thing, the cell membrane of muscle cell is called as sarcolemma. The cell membrane of muscle cell. The calcium containing pockets were actually endoplasmic reticulums. They are the sarcoplasmic reticulums. They are the sarcoplasmic reticulums of muscle cell. Okay. These red spots, red dots, these are nothing but oxygen carrying pigment inside muscle cells. We call them myoglobin. Like we have hemoglobin inside our RBC, the muscle cells or myocytes, they have myoglobin for carrying oxygen. So these are respiratory pigments, okay? They're red in color, and obviously high amount of these myoglobins are present into red fibers. Now when you talk about skeletal muscles, there are two kinds of skeletal muscles present into our body. One is white muscle, another is red muscles. Obviously red muscles needs more energy to perform their contraction, so they have more amount of more number of mitochondria as well as myoglobin. White muscles, they have less number of mitochondria as well as myoglobin. Okay. Now these parallelly arranged fibrils are called myofibrils. They are parallelly arranged and they have this dark light banding pattern, and you know that we call them, these light zones are called I band and the dark zones are called A band on them. Now how these myofibrils are attached with the sarcolemma or cell membrane of these muscle cells with the help of special proteins, and we call these proteins as dystropins. Okay. If there is gradual destruction of dystrophin proteins inside your body, then what will happen? You may face a disease called muscular dystrophy. Okay, so that's why the presence of these dystrophin proteins along with these myofibrils are very much important for our muscle cells. And here the invaginated part of cell membrane or their plasma membrane or sarcolemma are called as T tubules, and they are rich in ionic channels. Now let's move forward to the next part, that is myofibrils. Now we'll talk about a single myofibril present inside a muscle cell. So if you study a myofibril, you have taken out a single myofibril from a muscle cell, you will be observing banding patterns, right? So these light bands are called as I band and the dark bands are called as A band. And there is alteration of dark band and light band. There is alteration of light band and dark band. And that's giving the striated pattern of myofibril, and myofibril is giving striated pattern to the muscle fiber, and muscle fibers are giving striated pattern to the muscles. Okay, so these light bands are called as I band, and this abbreviation of I means isotropic band. So why we are calling them isotropic band? Because only thin filament, means this actin. Actins are called thin filaments, only thin filaments are present into this isotropic zone. So we are calling them I band. So that's why they will appear lighter under microscope. Now let's come to the dark band. This is called as A band or anisotropic band. Now why we are calling it anisotropic band? It's the entire length of thick filament. The length of A band is equal to the length of the thick filament or myosin filament, right? Myosin is called as thick filament and actin is called as thin filament. Actin filament is called as thin filament, myosin filament is called as thick filament. Now the entire length of myosin is actual entire length of A band, but why it's called as anisotropic band, a different reason is there, because it's written here overlap portion. Now what is overlapping here? You will find here myosin and overlapped part of, not full actin, part of actin. So that's why we are telling this. We have written here overlapped zone, right? So anisotropic, here you are getting myosins mainly. You are getting myosins and actin zone as well, means overlapped as well as non-overlap zone. So as because there is presence of more than one kind of protein, presence of myosin, presence of actin, we are naming this as anisotropic band. Now let's check the diagram in a proper manner as given in NCERT. So this is a single myofibril. Now upon this myofibril, we can separate out different zones. So this particular zone can be called as A band. This will appear dark. And this particular part is called as I band, which looks comparatively lighter in compared to A band, and A band is again surrounded by I band. So it's alteration of A band and I band. Okay. Now let's focus the middle part of I band. Focus the middle part of I band, you will find this zigzag strip of disc or protein that is called as Z disc. At the middle of I band, means I band is bifurcated by a zigzag disc or zigzag protein strip that is called Z disc, and the length in between one Z disc and another Z disc, the length in between two Z discs, you can call it as one sarcomere. The length between two Z discs are regarded as one sarcomere, that is the unit of contraction of muscle fiber. Unit of contraction is one sarcomere, and repetitively multiple sarcomeres are present here. Okay. Now we will be enlarging one sarcomere to study in detail. The first I said that there is a zigzag protein strip called Z disc. What is the importance of that Z disc? With that Z disc, you will be finding these actin filaments are associated with. So, actin filaments are anchored with Z disc. And these black strips are myosin filament. This myosin filament, the central part of this myosin filament, you will get another band of protein, another disc of protein that is called as M line. So we can say that the length of A band or the length of myosin filament is bifurcated by M line. So Z disc bifurcates the length of I band and M line bifurcates the length of A band. So M line and Z line and their importance. M line present at the middle of A band, middle of A band, that means that is bifurcating the length of A band. And when you talk about the Z disc, that is bifurcating the length of I band. It's present at the middle of I band, or you can say it's bifurcating the I band. Sarcomere, the unit of contraction, and also can be said as the distance of two consecutive Z band in between two Z discs. The place in between two Z discs are called as one sarcomere. Now, if you consider one sarcomere, how many A band you will be finding inside it? One complete A band you will be finding, and two half I band you will be finding, because the Z band is bifurcating the I band into two half. One half is associated to the next sarcomere and another half is associated with this particular sarcomere. So that's why half I band into right side, half I band into the left side. So one sarcomere is made up of one A band and two half I band. Very important question. That is, one complete sarcomere is made up of one complete A band and two half I band. So the length of myosin, the length of myosin is our A band and only actin zone is our I band. only actin zone is our I band. Now, who is bifurcating the I band? Z line is bifurcating the I band. Z line is bifurcating the I band. Here one Z line, here another Z line. And the zone in between the Z line, that is called one sarcomere. So, if you look very closely, you can observe this I band is bifurcated into two halves by this Z line. So, only half of the I band is falling inside one sarcomere, but a full A band is falling inside the sarcomere. And this A band is bifurcated by M line. And you can observe one more thing, the zone here, the middle part of the M line, middle part of this A band near to the M line, a particular distance that has only myosin, no actin. It is called non-overlap zone, and a particular length of A band, you will be getting myosin as well as actin. You will be getting myosin as well as actin. So that is called as overlapped zone. So I am making it more clear by another diagram. Okay. Suppose this is the M line and these two are Z lines. Now, M line will be associated with or present in the middle of these myosin proteins. These are thick filaments or myosin filaments. So total length of thin filaments are called as A band, right? And A band is bifurcated directly by what? A band is bifurcated by this M line. That's also very much important thing to remember. Now let's attach the actin proteins with Z line. Attaching the actin filaments with Z line. So you can say that length of actin is bifurcated by Z lines as well. And this particular zone is called as I band, where only actin is found. And beyond that, another A band will be started. Myosin filaments are there. Okay. So now this is a perfect sarcomere formation can be seen here. So sarcomere, I am talking about the sarcomere, the length between two Z lines, that is one sarcomere, the length between two Z lines. Now it has one complete A band. Now I band is bifurcated, this non-myosin, only actin zone. Only actin zone is I band. Right? And that is bifurcated by the Z line into two half. So we can say that for a single sarcomere, half I band from left side and half I band from right side, both of them are involved, and a full A band from the central part is involved, right? Now here let's check the A band in a proper manner. The middle line is called M line, but here you will be getting a part where both myosin and actin is overlapping each other. Myosin and actin, they are overlapping each other. And there is only myosin zone in middle. Only myosin zone in middle, there is no overlapping of any actin. So here comes a new story. Inside A band there is overlap zone and non-overlapped zone. Yes. So this non-overlapped zone is known as H zone. Non-overlap zone is known as H zone, where you will be getting only myosin fibers or myosin filaments inside the A band, and zones with actin and myosins are called as O zone or overlap zone. Now under microscope, the O zone will appear darkest, H zone will appear comparatively lighter or darker overall, and the I band will look lighter. So the darkest zone is overlap zone of A band, darker zone is H zone of A band, and the light zone will be I band of a myofibril. So we are moving forward to the next topic, the next part of this particular topic. So we know few things, that what is sarcomere, what is M line, what is Z line, what is A band, what is I band, how a sarcomere is made up of. A sarcomere is made up of one single A band and two half I bands, right? Now inside one single A band, if you talk about one single A band has a non-overlap zone that is called H zone, that is present in the middle of A band surrounding the M line and has two overlapped zone, or you can name them O zone. Into this H zone you will be finding only myosin, but into this overlap zone you'll be getting actin plus myosin. Z line bisects or bifurcates I band and M line bisects A band, or it can be said as the A band is total length of myosin filament. So M line can bisects the length of myosin filament or thick filament. Now let's get inside the structural, the biochemical aspect of these myofilaments. So when you talk about the proteins present inside the myofibrils, few will be contractile protein, few will be regulatory protein, and few will be structural protein. Okay, who are contractile protein of muscle fiber? Actin and myosin, they are called as contractile protein, where actin is called thin filament and the thickness is 8 nanometer. Myosin is called thick filament and the thickness is almost double, that is 16 nanometer. Okay. Now this myosin filament, so these proteins are called as motor proteins. Why they are called as motor proteins? During the time of muscle contraction, the utilization of ATP will be by the myosin proteins. So the proteins which are responsible for utilization of ATP, they are called as motor proteins. So basically these myosin proteins are responsible for conversion of chemical energy into mechanical energy by hydrolysis of ATP into ADP plus PI, and the energy that is released used by the myosin for the muscle contraction or the motor movement. That's why in between actin and myosin, myosin is called as motor protein, though both of them are contractile protein because they are responsible for muscle contraction. Basically what happens during the time of muscle contraction, actin and myosin binds with each other and myosin will be pulling the actin towards the center, that causes muscle contraction. Okay, and as regulatory protein we got the names of tropomyosin and troponin. Now why these proteins are called as regulatory proteins? Because they will be deciding when and how the myosins will be binding with the actin or not. They the deciding factors, troponin mainly, along with tropomyosin, that the myosin protein, so the head of myosin, will they be binding with the actin or not? So that is called as the masking and unmasking phenomena. They are responsible for masking effect of actin filament. So that's why we call them regulatory proteins. Now there are some few more proteins associated with your muscle. They are titin. They are the largest among proteins present into muscle. Dystrophin. Without these proteins, you may face severe loss of contractility. Alpha actinin and myosin. So these are the types of proteins you will be finding inside your muscle fibers. Okay. Now let's start another topic, that is structure of contractile proteins. So you know, these thin and thick filaments, actin and myosin, they are considered as contractile proteins. So first we will talk about actin. So the monomers of actins are called as G-actin. They are globular proteins. With the help of ATP and magnesium ion, they will polymerize, and upon polymerization, these globular actins will form filaments. We call them F-actin or filamentous actin. And the next step, filamentous actins, two filamentous actins will wrap each other like snakes, will get will form a helical structure. Two filamentous actins will form helical structure like two snakes are holding each other, and will form the actin filament. Okay. So inside one actin filament, how many F actins are there? You need two F actins to form a single actin. Okay. So now we are not completed yet. In while we are talking about formation of the thin filament, we need to wrap them with the regulatory proteins. Okay. Now this green color filamentous proteins will be wrapping the active sites on actin filament, and we call them tropomyosin. But these tropomyosins cannot sit alone on the active sites or cannot bind alone on the active sites on actin filament. They need help of troponin proteins. So these are troponin. Now it's completed the formation. When you're talking about formation of thin filament, two F actins, two tropomyosins, and many troponin trimers. Okay. Now let's talk about the troponin, which is responsible for the masking of actin mainly. This is the protein responsible for masking of actin. The active sites present on actin. So they are basically trimers. They have three subunits, and we call them TPT, TPC, and TPI. Let's talk about their abbreviation. TPT. With this particular subunit, this troponin molecule will bind with tropomyosin. So responsible for tropomyosin binding. Now TPI and TPC. What do you mean by this? TPI, with this subunit, that will be inhibiting the binding of myosin. So this is inhibitor site, inhibitor of myosin. TPC, with this particular subunit, it will bind with calcium ion. So it's calcium binding site. Now we have come to that topic that is the most tricky and the conceptual one of this chapter, that is the mechanism of muscle contraction, and the theory was given by two German scientists, Huxley and Axley, and the theory is called sliding filament theory. Yes. So, what is the basis of sliding filament theory? They said that when there is muscle contraction in our body, the filaments, means actin and myosin, basically the thin filament is sliding over thick filament. The thin filament is sliding over thick filament. So just think like that. One person is quite healthy, a bodybuilder, and one person is suffering from malnutrition, very thin and lean person. So that thin person can ride the shoulder of the bodybuilder. So that is quite easy phenomena, quite possible phenomena. But if opposite occurs, then what will happen? The thin person or the lean person will get collapsed, right? So similarly, we know about two filaments, that is actin, the thin filament, and myosin is the thick filament. So obviously, when they will be sliding past, past each other. So it's the myosin or the motor protein whose position will be in the below of the actin, and the actin or the thin filament will slide past thick filament. Got it? So how it starts? It starts with a motor signal which comes from your central nervous system, the processing unit of a body. Okay. And this motor signal will be passed into the muscle fiber with the help of motor end plate. This is the neuromuscular junction. Motor end plate is nothing but the neuromuscular junction. And that will cause release of neurotransmitters acetylcholine, right? Now the acetylcholine will be released from the motor neuron presynaptic knob, and they will bind with the acetylcholine receptor present on the surface of the skeletal muscles, present on the receptors of sarcolemma, and they will initiate opening of sodium channel, and that will cause depolarization of sarcolemma. Now what do you mean by depolarization of sarcolemma? Sarcolemma will get converted from negative to positive. So that is called depolarization. And when it's getting depolarized, there will be generation of action potential throughout the sarcolemma of the muscle fibers, and that will trigger the release of calcium ions from sarcoplasmic reticulum. Okay. Now we need this calcium ion. The cell will order this calcium ion, please go and bind with the troponin, as because these troponin molecules, they are responsible for masking the actin. Now these troponin molecules, they are very much greedy or hungry for the calcium ion, whenever they will see free calciums are hovering around, so they will jump and go to catch the calcium to bind with them, and in the meantime tropomyosin is also get displaced from the position, and the actin and the binding site, the active sites present on the actin filament, they are opened for the myosins, and that will trigger actin myosin binding. Okay, so release of calcium from the sarcoplasmic reticulum will trigger calciums to bind with the troponin molecules, and that will trigger dislocation of troponin along with the tropomyosins, and that will be causing unmasking, of myosin binding site on actin, and the next moment the myosin head will hydrolyze the ATP. It was waiting to hydrolyze it. It needed just needed a signal, and that signal is the unmasking of active sites present on actin. Now, in hurry, myosin head will hydrolyze the ATP molecule. Now, what do you mean by hydrolysis? ATP will be broken into adenosine diphosphate and inorganic phosphate, and that will release energy, and that energy will be harvested by myosin head, the motor part, and there will be cross bridge formation, means the binding of myosin with actin is called cross bridge formation. Again I am repeating, binding of myosin head with actin is called cross bridge formation, and upon the formation of cross bridge, now why actually myosin wanted to bind with the active side on actin? It's gonna be like it will exert the energy just it has acquired for the process of pulling or sliding the actins over the thick filament. So when the myosin filaments are pulling actin, that will cause sliding, and towards the direction of the M line, the myosins are holding the actin filaments and pulling towards the central line or M line, and that will cause shortening of sarcomere, and if sarcomeres are shortened, then the length of total myofibrils will be shortened, and if the length of myofibrils are shortened, the length of muscle cells as well as muscle will get shortened, and that will cause contraction of muscle, right. So here, as given in the NCERT, we will be discussing this particular phenomena of muscle contraction under sliding filament theory. Okay. So let's start here. A myosin head, it needs ATP. Now to study that, we need to study the structure of myosin in detail. Yes. First, study the structure of myosin. So here we know that the monomers of actin are G-actin. In case of myosin, the monomers are meromyosin. Hundreds of meromyosins together will be forming thick filament or myosin. Now let's discuss the structure of one single meromyosin. The structure looks like a golf stick. This is structure of one meromyosin molecule. This part is called LMM, light meromyosin, or you can call it tail of meromyosin molecule. This part is called as globular head of meromyosin, and this particular part is called as short arm or hinge of meromyosin molecule. Yes, and together this head and short arms can be called as cross arm. Do not confuse between short arm and cross arm. So cross arm equals to head plus short arm, and the another name of cross arm is HMM or heavy meromyosin. So the heavy meromyosin part has globular head as well as this short arm, right? Now let's focus on this globular head. This globular head is nothing but acting as ATPase enzyme. The globular head is acting as ATPase enzyme, so it has the substrate binding site. So here you will be getting two sites, one ATP binding site on each globular head and one actin binding site. So the green circle, that is actin binding site, and this purple circle, that is ATP binding sites. So these are the active sites present on these ATPase head of meromyosin. With this actin binding site, it will bind with the active site present on the actin, and with this ATP binding site, it will catalyze the reaction of ATP hydrolysis. So when we are talking about meromyosin, it has two parts, HMM heavy meromyosin, LMM light meromyosin. Heavy meromyosin has a globular ATPase head and a short arm. The ATPase head has two active sites or binding sites, that is ATP binding sites and actin binding site. The short arm is if you are comparing the structure with a golf stick, so this angle formation part, this part is forming the angle, that is called as the short arm part. Light meromyosin, this is called the tail portion of a meromyosin. Now let's come to the sliding filament process. So this is our meromyosin molecule, the thick filament meromyosin head. It has the ATP binding site. So suppose the ATP is already there, like ATP already binds with the ATP binding site present on the meromyosin head. So the next step, it will be hydrolyzing the ATP. It will be ATP hydrolysis, and it will form ATP is splitted into ADP and PI, adenosine diphosphate and inorganic phosphate, right? Now, upon the process of hydrolysis, the myosin head will harvest the energy released during the time of this ATP hydrolysis, and that energy will be acquired by the myosin head. It will get charged and it will form a 90-degree angle to go and bind with, to show cock and bind with the actin filament, the binding site, the active site present on the actin filament, and that is called cross bridge formation. This is the myosin head. So, during the time of cross bridge formation, you can find that the angle of the myosin and the myosin head, the angle is 90-degree or right angle. Okay. Now, it's not, the process is not ended here. What after cross bridge formation, the myosin head is charged with energy, with kinetic energy. Now it will convert the energy into mechanical energy and it will give a power stroke or pull the actin fiber towards the M line. It will pull the actin fiber, towards the M line, and that is called the process of sliding or rotation or power stroke. And when it is done with pulling or sliding or rotating the actin filament over the thick filament, then it is exhausted, then it will throw the products, that is ADP or inorganic phosphate, out of the myosin head. So, you know this simple enzymatic reaction. First enzyme will come closer to the substrate, then it will form enzyme substrate complex, then it will form enzyme product complex, then enzyme and products are separated and the enzyme is ready for another catalytic action. So, when the ATP is binding on the active site of myosin head, so that is forming enzyme substrate complex. When ATP is broken into ADP and inorganic phosphate, that is forming EP or enzyme product complex, and in as AP state, enzyme product complex state, it's forming cross bridge and it's performing the sliding. After performing the sliding, it will be releasing the product like ADP and inorganic phosphate out of the myosin head, and now this present at E plus P format after power stroke or after sliding. Now it's time for binding of a new ATP. A new ATP will come closer to the myosin head and that will stimulate breaking of the cross bridge, and it will hydrolyze ATP and it will bind with another active site present on actin and it will perform another sliding. So, here one very important question is lying in between these two steps. After performing the sliding, the myosin head is releasing ADP and inorganic phosphate from the active sites. Now, until unless it's not getting new supply of ATP, the cross bridge won't be broken. So, question is asked, which of the following factor or following phenomena is responsible for the breaking of cross bridge? It's the attachment of a new ATP at the actin binding site of myosin. When a new ATP will come and bind with the active site, then only the myosin will be loosen the cross bridge, and if there is no ATP supply, the myosin head will keep on like in a static condition binded with the active site on actin. Remember this. So let's sequence-wise study the phenomena of muscle contraction, starting from the relaxed state, then initial contraction and the maximum contraction. Okay. Now you have to take the reference of the structure of myofibril and the myofilaments. Already we had studied that. Okay. So you have a basic idea of I band, A band, H zones. So you can observe here, when a muscle is relaxed, A band is maintaining the length, I band is also maintaining the length, H zone has its own length, sarcomere has its own length. Now, while started contracting, the initial contraction, what you can observe here? So it's observed that the I band is getting reduced in terms of length, but there is no change in the length of A band. There is no change in the length of A band. There is no change of A band because there is no change of length of myosin fiber. And even the I band is getting reduced, but there will be no change even in the length of individual actin filaments. You need to remember this. So during the time of muscle contraction, actin filaments will slide over the myosin filament. So that's why there will be reduction in the length of I band, but there is no individual reduction in the length of myosin filament or actin filament. Okay. Now, while gradually the actin filaments are sliding inside or sliding inwards towards the M line, then what will happen? The A band will cover more overlap zone, and if there is more overlap zone, the length of H zone will also be reducing. The length of H zone will also be reducing. The length of H zone will also be reducing, but there is no net length change of A band, right? Now let's observe the muscle contraction for maximal contraction. When maximally contracted, the actin filaments totally overlapped the A band or the myosin filament. So H bands are obliterated or vanished. H zones are obliterated and vanished. Z lines are touching myosin filaments. So I bands are also obliterated and vanished. So here I wanted to tell the important questions can be asked based upon this particular topic. Which all bands or which all structure present into the sarcomere or into the myofibrils can be reduced, cannot be reduced in length, or can be vanished. So first we'll talk about the structures which cannot be reduced in length. They are A band, actin and myosin. These structures cannot be reduced in length. Next, can be reduced or shortened in length. and the structure are I band can be shortened, H zone can be shortened or length can be reduced, sarcomere, for sarcomere the length can be reduced. For all this structure there is possibility of shortening. Obviously there will be shortening during the time of contraction. Okay, now during the time of maximum contraction they can be vanished or obliterated. They can be vanished or obliterated during the time of maximal contraction. During the time of maximal contraction, this particular zones can be vanished or obliterated. I band can be obliterated, H zone is obliterated during maximal contraction. There is no I band, no H zone, because the entire length of A band is occupying the length of sarcomere. So at maximal contraction, it's possible that the length of sarcomere equals to the length of A band, or the length of sarcomere at maximal contraction equals to the length of myosin filament. That is possible. Okay. Now let's move to the next topic of this chapter, that is differences between red fibers and white fibers. So while we are differentiating between the red and white muscles, let's take the points. Presence of mitochondria, myoglobin and sarcoplasmic reticulum. So for red muscles there will be more myoglobin and mitochondria, but less sarcoplasmic reticulum. For white muscle there will be less abundance of myoglobin and mitochondria, but more or high abundance in sarcoplasmic reticulum. Now what is the science behind the presence of more mitochondria and more sarcoplasmic reticulum? See, red muscle fibers are called as slow twitch muscle and white muscle fibers are called as fast twitch muscle, means faster contraction. Red muscle fibers are responsible for power generation, endurance, sustained contraction, but white muscle fibers, they are responsible for power generation, responsible for powerful contraction. Red muscle fibers mainly will be relying upon aerobic respiration, so these are called as aerobic muscles. White muscles is because they have less number of mitochondria into them, they are called as anaerobic muscles. Okay. Now why they are contracting very rapidly? Because they will get fatigue very fast. So they are fast fatigue muscles. But as because they are responsible for endurance or sustained muscular activity, they are slow fatigue muscle. Now examples can be asked. Now suppose for runner, if someone is running into marathon and another is running into just 100-meter race or 200-meter race. So the marathon runners, they need prolonged muscle contraction, so they will have red muscles into them. But those who are running 100-meter race, 200 meter race like Usain Bolt, he has mainly white muscles into his limbs. Okay. Another example, the flight muscle of sparrow and eagle. Now if you compare them, sparrow will fly into a short distance and it's a very quick phenomena. So it needs first twitch. The flight muscle of sparrow is white. But the flight muscle of eagle or vulture, they're hovering to the high altitude for prolonged duration, searching for their prey. So they have red muscles. Flight muscles of eagle or the muscles of sprinters, means who are running for 100-meter race or 200-meter race, they are called sprinters, have white muscles as well as whereas marathon runners, they have red muscles. Okay, so we are almost done with muscles. So here you can see the skeleton of our friend Chintu. He is taking a selfie after the class, but obviously the movement of his body is due to the presence of the muscles associated with the skeleton. If there is no muscle associated with the skeleton, there will be no movement into the body of Chintu. Let's make 6-pack, 8-pack abs. So either you are dancing or you are showing movement, it's the muscles present into your body along with the skeleton. Okay. Now we'll talk about the disorders associated with muscular system, and we need to talk about two disorders. First, myasthenia gravis, and secondly, muscular dystrophy. Okay. Myasthenia gravis and muscular dystrophy. First, talk about their cause of disease. So, myasthenia gravis, this is an autoimmune disorder. Where your own antibodies, own defective antibodies are attacking your own cells, your own neuromuscular junctions, right? So when you're talking about myasthenia gravis, we need to talk about motor unit of our body. So when you are studying motor unit, it starts with a motor neuron. This is the presynaptic area of this particular motor neuron. And is forming neuromuscular junctions with muscle fibers. So, the associated muscle fibers, the muscle cells, along with the neuromuscular junctions or motor end plate, can be also named as neuromuscular junction. The motor neuron, the motor end plates, and the muscle cells. Together we will be calling this as motor unit, and obviously there will be release of acetylcholine to transmit the signal, motor signal, into the muscle cells for the creation of action potential and initiation of muscle contraction. For that, these muscle cells, they have special receptor for acetylcholine. Now, if your antibodies will destroy this receptor, acetylcholine won't be binding with this muscle fiber or muscle cells. So, neuromuscular junction is affected, and that we call myasthenia, means weakening of muscle. Asthenia, my means muscle, asthenia means weakness. So, this weakening of muscle caused by autoimmune disorder that will destroy neuromuscular junctions of your body. Okay. Now, muscular dystrophy. This is progressive degeneration. This is a genetic disorder. Here what happens? Progressive degeneration of your muscle due to degeneration of dystrophin protein. The myofibrils will losing their activity. And the causative factor is genetic disorder. So, we are done with muscular system. Now, we will solve few questions and we will end this one shot of this particular chapter, particular topic. Okay. So, there is a statement is written here, fascia, that is a loose connective tissue which attaches fascicles. So, if you read very superficially, you will think this is a true statement, but there is a mistake lying inside it, that is the loose connective tissue. Fascia are made up of dense connective tissue, dense irregular connective tissue, they are not loose connective tissue. So, that is a false statement. So, statement 2 is saying that each organized skeletal muscle in our body is made of number of muscle bundles or fascicles. That is true statement. So, we can say that statement 2 is true, but statement 1 is false. Next question, ATP is found in the myosin head, the globular head of myosin. So, that is the answer. Okay. Third question, find the odd one in terms of the type of movement. Fallopian tube, you will be finding ciliated epithelium, ciliary movement. Respiratory tract, yes, ciliated epithelium, ciliary movement. Paramecium, again ciliary movement. Spermatozoa, flagella, flagellar movement. So this is the odd one and our answer. Now let's come to the next question. X is a muscle associated with eye, great, which help you to rotate your eyeball. That means obviously it's a voluntary muscle. The muscles present inside your eye, that is iridial muscles, they are involuntary, but these muscles associated with your eyeball, they are voluntary muscles, which is not the character of that muscle X. Okay, so that means voluntary means striated muscles. It is striated, it is the character. It is involuntary, this is not the character of that particular muscle. It is cylindrical in nature, that is also correct. It is unbranched, that is also correct. Now let's follow next question, assertion and reason. Assertion is saying Hydra can use its tentacles for capturing its prey and also use them for locomotion. That is true. Reason is saying that the two may be linked by stating that all locomotions are movement, but all movements are not locomotion. Reason is also true, but can we connect them? Hydra use its tentacle for capturing prey and also use them for locomotion, because all locomotions are movement, but all movements are locomotion. It is not going with each other, though both the statements are correct. So the answer will be both assertion and reason are true, but R does not satisfy assertion. Okay, next assertion, another question. Locomotion requires a perfect coordinated activity of muscular, skeletal and neural system. A true sentence. And the reason, the sentence is like that: the skeletal muscles are excited upon threshold stimuli from neural system and transmit the force generated through the skeletal system and joints. That is also true. So there is perfect reason. So why we are saying that locomotion is a coordinated activity of muscular, skeletal and neural system? As because the neural signal will come, then it will stimulate the muscle contraction, and the force generated by muscle contraction. Now you know that muscular system responsible for generating force for movement, locomotion and also for production of heat into our body. So the force generated by muscle contraction will be transmitted through the joints of our skeletal system. So both assertion and true, and reason satisfied the assertion. Okay, so with this I am ending the particular topic, that is muscular system of the chapter locomotion and movement. And I am also coming with the next topic, that is skeletal system. Till then, stay tuned. Bye bye. Jai Hind.